Yeah. Yeah. Good afternoon, students. Welcome back to Botany second year class. So I hope that everyone has gone through these classes, and who gone through these classes, they might have prepared till today all the SAQs and the VSAQs and the LAQ. At least one LAQ if you prepare, second LAQ half also you will get 4 marks, does not matter. Okay, so here 60 out of 60 or above 50 is not the target for us, isn't it? We need to pass through and get at least first class mark, that is above 45 are mandatory stream what we are making, that is the passing marks. If you think, then only you will prepare well. Otherwise, if you prepare passing marks for 21, then you will be trying to get only 30. You won't get the 50, that is the 45 marks where first class you will get it. Okay, target in such a way, the target always should be more. Then we will try to achieve the target at any cost. So our willpower and all our efforts will be dedicated to that target, to reach the target. Okay, so be prepared like that. In your future also you have to do like this. We have to aim for the betterment. Okay, always for a bigger ones, not for the small ones. So here we have to see the VSAQ part, isn't it? We have done the VSAQs from the 9th chapter we have finished, and the 10th chapter we need to do, where 2 VSAQs we will get. So now you can check out your material, whatever you are having, so that what I am saying, these are the in-text questions. From this only you will get the 2 VSAQs from the 10th chapter. That means if you read the nearly 20 questions, 4 marks you will get. That's a plus point. Profitable in the business wise, if you think, that's a profit always. Okay. So here, what we have to see in this also, most important ones. Distinguish between heterochromatin and euchromatin. And which of the two is transcriptionally active. Euchromatin, the regions of the chromatin which are loosely packed and light stained, those are the euchromatin. Euchromatin means loosely packed and lightly stained. Lightly stained chromatin material is called as the euchromatin, which is genetically, transcriptionally very active. Okay, which is transcriptionally active. Now we have to see the heterochromatin. What is meant by heterochromatin? Heterochromatin. Heterochromatin means where the chromatin material is densely packed. Where the chromatin material is densely packed and darkly stained. And transcriptionally, it's the weak, which cannot do the transcription process. That is the heterochromatin. This much you have to write over here, so that we will get 2 marks very easily. Because this is the, in the syllabus also we learned this part during this type this lesson itself. Okay, and the next one is on. That is the Hershey chase and in the bacteriophage. Hershey chase. These are the scientists who has proved the DNA is the genetic material in which organism? Bacteriophage. And here, this bacteriophage is a viral organism. And this one, you have to write it as BACTERIO, bacteriophage, PHAGE. It's not a PHASE. If you are writing SE, then you will lose the marks. Okay, make sure this. And the next one. What are the components of nucleotides? Nucleotide. Nucleotide means nitrogen base plus nitrogen base plus pentose sugar plus phosphate bond. This is called as the nucleotide. Easy. Okay. And the next one. Given below is the sequence of coding strand of DNA. This is the coding strand of DNA in a transcription unit. Okay. So, right, what they are asking for the first bit, A bit, they are asking the complementary strand. Okay. So, what they have given from the 3' to 5'. What they have given AAT, GCA, GCT, ATTA, GG. This is the 5'. Okay. Otherwise, I'll write here. It will be easy. My test stretch I can show you. A, T, G, C, A, G, C, T, A, T, T, A, G, G. This is the 3' to 5' end. So now answer I'm writing in a different color. See. For this one, what we have to write? First of all, the complementary strand. Complementary strand means second strand for the DNA. So that has to start with the 5' to 3' direction. Isn't it? This is one strand. This is one strand. Okay. Now, this is another strand. What we have to write now? Complementary to it. A always make a bond with the T. So, T, T, A, C, G, T, C, G, A, T, A, T, C, C. This is the strand what we have we will get with the complementary. Complementary means which is in the opposite direction to the first strand, okay, of the DNA. If this is the 5' to 3' strand, then this is the 5' to 3' in the opposite direction. So like this, whenever two strands are there, one is the template, another one is the complementary. Complementary strand will have the opposite bonds now. Like that, if you write it, it will be easy. And how to read this? Okay, don't take much more time in reading and writing the nucleotides over there. When we are here, I can show you with the different color. Okay, on the question paper also, you can mark like this, how I am doing right now. So see here, AAT, 3 nucleotides at a pair. Okay, at a stretch, if you can do, then it will be the best one now. So AAT is one, GCA is one, GCT is one, ATT is another, and the last one is the AGG. Then we can write like that the codes for the complementary strand. Okay, and the next one is the what we have to write in the second bit. They have given the second bit from the 5' to 3', and the mRNA strand we have to write for this DNA strand. So what is the DNA strand? The nucleotides on the DNA strand, AAT, GCA. Now again GCA. After that, GTA. Okay. Next one, TTA, GG. That is the 3' end. So the mRNA is going to be having same ends beta. In the previous answer, I have written the codes on the strand in the 5' to 3' direction when the strand of the question is starting from the 3' to 5'. If you remember it well. Okay. So now on the mRNA, whatever may be the ends that are there on the DNA strand, same strand as it is, we need to take the ends over here in the answer. We should not use the another pattern, that is the 3' to 5'. Same. So in this one, wherever T is there, replace the T with U. That's it. All others are the same. Okay. T is there means U. So in this also, I am saying, see, 3 at stretch if you have taken, then that will be the easy one. And in this also, underline the T. Then we can easily say whatever our answer is there. And this also same thing. Okay. AAU. Next GCA is same. No change. And this GCA is also same. No change. Next GTA is there in the question. That is GUA. Understood? I missed something. Right. AAT, GCA, GCA, CTA. Yeah, right over. GCA, GCU. They have written wrong. Okay. And GUA. Now UUA. Here GG will be coming. Okay. Like that, we have to write the mRNA strand codes. Okay. When we are writing like this, see here only T is going to be replaced with U and all other codes are as it is. That's the mRNA strand. If you remember this much, it's sufficient for the, and this we can expect in the exam. Next one. Name any two viruses, and whatever I'm marking, just mark in your books also, stress on this VSQs. No chance for you to use your intelligence right now. So believe us, we won't misguide you. Okay, name any three viruses which have RNA as the genetic material. So in this, we can write only the organism's name. TMV, BZ1 and HIV is easy, and poliovirus is also easy, isn't it? And the QB bacteriophage, if you remember that, you can write otherwise three. They asked three, we have given three organisms. Done. Okay. Otherwise, this much only. Easiest one. And coronavirus also you can write. But apart from these three, that should be there. If spelling was gone wrong, then we are not responsible. You have to. What are the components of the transcriptional unit? Transcriptional unit is having three components in it. That is the promoter, and the structural gene. Structural gene. And the last one is the terminator. Promoter, structural gene, and terminator. So three will be there as the components. So these three components we have to write as the transcriptional unit. Next one, what is the difference between the exons and introns? Exons and introns. When you are writing this, make the tabular column like this. Exons and introns. Okay, like that if you write, that looks very pretty, the answer. Okay, exons means, exons are the coding sequences. Exons are coding sequences. Coding sequences, okay, that appear in the mature processor mRNA. You can write as the second point. First point is the exons are the coding sequences. Introns are non-coding sequences. Non-coding sequences. Now the second point. Okay, the exons will appear in the mature mRNA. Appear in the mature mRNA. And introns will not appear in the mature mRNA. This much we have to write. Easy to write these differences and easy to remember also. What I think so. Because usually I'll say the easiest methods to remember and to write. And this question is also important one. Differences between exons and introns. Next one. What is meant by capping and tailing? What is meant by capping and tailing? Okay, Capping means on the HN RNA, HN small letters, R, N, A capital letters. Okay, remember if in this, if you have not written the capital letters in the RNA, marks will not be given properly. That represents the another meaning. So enzyme, it's not a genetic material. So we cannot accept it. So be sure that RNA will be in the capital letters. HN should be the small letter, case sensitive over here. If you won't follow, then we need to follow the rules and regulations a lot where you won't get the full marks. Okay, capping means and this capping and the tailing parts are present on the HN RNA. Okay, so the HN RNA undergoes the additional processing called as the capping and the tailing part. So capping means in the capping, the nucleotide will be present at one end, that is the methyl guanosine triphosphate. Okay, methyl guanosine. If you cannot able to write this, I will say the easiest method. Methyl guanosine triphosphate. Okay, MGPPP, triple P. Otherwise, TPP. Anything. Triphosphate. Okay, methyl guanosine triphosphate. This one should be written like this. Methyl guanosine triphosphate is added at the 5' end of the HN RNA. Okay, capping means to the HN RNA at the 5' end, a protein is going to be attached called as methyl guanosine triphosphate, that is MGPPP. Okay, like this also you can write. Next one, tailing. Tailing means it is present at the another end of the HN RNA. If capping is present at the 5' end, tailing will be at the 3' end. It's mandatory. So at the 3' end of the HN RNA, a template independent, that is the adenylate residues are going to be added up over there. Okay, that is the tailing part. Adenylate residues are present at the 3' end. If you have written this much, it's sufficient to get 2 marks over here. And the next one is what is meant by point mutation? Give an example. Point mutation. Point mutation means the mutation which occurs at a single gene on a chromosome, otherwise at a single base pair of the DNA is called as the point mutation. Example, sickle cell anemia. Example, sickle cell anemia. What is meant by point mutation? When the mutation occurs due to the change in mutation occurs due to the change in a single mutations occur due to the change at a single nucleotide base of DNA or a small part of a small part of gene is called as point mutation. Okay, so this point mutation, best example, sickle cell anemia. And the next question. What is the function of codon AUG? What is the function of the gene code AUG? AUG is the starting codon and which also represents the enzyme methionine, amino acid methionine. If you write that, it's sufficient. Okay. What are the functions of AUG? AUG is a starting codon. It's a starting codon as well as it represents the amino acid methionine. These are the two functions of the AUG. That's it. Is having the dual functions. Only this code is having the dual function. And the next one, define stop codon. Write the code. Okay. Stop codons means the codons which are not representing any amino acid, otherwise stopping the process of translation, that is called as the stop codons. Okay, stop codons means the codons which stop the translation process, stops the translation process, that is the polypeptide chain synthesis, or else which never code for the codons which stop the translation process are called as the stop codons. These stop codons will not code for any amino acid. These stop codons will not code for any amino acid. Those are the stop codons. They are asking to write the codons also. Then that is the UAA. There are 3 in number. 3 stop codons you have to write. UAA, UAG, UGA. These are the 3 stop codons. Okay, those are present in the genetic material for the transcription process. So need to be written properly, stop codons. And the next one is, what is the difference between the template strand and the coding strand? What is the difference between template strand and the coding strand in a DNA molecule? Template strand and coding strand. What they mean? Okay, template strand means the strand which is running from 3' to 5' direction. The strand which is running from 3' to 5' direction is called as the template strand. Okay, and the another strand which is running from 5' to 3' and anti-parallel to the template strand is called as the coding strand. Okay, and this coding strand is having the what we can say, this during the transcription process, during the transcription process, the sequence of RNA is can be guessed by the coding strand codes. Okay, and in that the thiamine should be replaced with the uracil. That's it. What we have already seen in the template strand and the code complementary strand and the coding strand we have seen now. Same thing. And the next one is chemical differences between the DNA and RNA. Any two chemical differences they said. At that time, you should not say what you know, whatever you feel free. Okay. So here DNA means deoxyribonucleic acid. And in this, right, deoxyribose sugar is present. Okay. Deoxyribonucleic acid, we can say na DNA. DNA means deoxyribonucleic acid. Right? Once we are saying this, which type of sugar it is having? It is having the deoxyribose sugar. Okay, this is present in the DNA. Then it is having the nitrogen bases are adenine, thiamine, cytosine, and guanine. These are the 4 nitrogen bases that are present in the DNA. Whereas the RNA is having ribose sugar in it. What it is having? It is having the ribose sugar in it. And the nitrogen bases are adenine, uracil, guanine, and the cytosine. Instead of thiamine, you have to write the uracil over there in the nitrogen bases. That's sufficient for the RNA. Okay. Next question. In a typical DNA molecule, the preposition of thiamine is 30%. 30% thiamine is present in the DNA. What they are saying? 30% thiamine of the nitrogen bases. Find out the percentages of other nitrogen bases. So in the DNA, thiamine, what they have given? Thiamine is of 30%. So what about the adenine, guanine, and the cytosine? So always, how many thiamines are there? Those many adenines need to be there because they will make a bond. So adenine is also of 30%. See here, I am writing the answer. Okay, adenines are of 30%. And in the 100, 30 plus 30, 60 were over. How many remain? 40 now. So in that 20 should be guanine and 20 will be the cytosine. Done. Because we have to follow the Chargaff's rule. If I'll say this Chargaff's rule, everyone may not take interest. But here A plus G is equals to C plus T. How many purines are there? Those many pyrimidines should be there. Okay, that is the Chargaff's rule, which equivalents to 1. So in that case, this is the way how to make the calculation. If you don't know this also, blindly you can follow what I said. Okay, what I said, blindly follow. Always adenine makes bonding with thiamine and guanine makes bonding with the cytosine. This is constant one. So in that case, those many adenines should be there. Same quantity of guanines and the cytosines should be there. Okay. So like that, we have done the calculation part. Now comes the second type of question. The preposition of the nucleotides in a given nucleic acid are Adenine are of 18%. Adenine are of 18% what he has given, isn't it? And guanines are of 30%. And cytosine of 42 and uracil of 10. Now he is asking the question, see, name the nucleic acid and mention the number of strands in it. So, in which nucleic acid, in which genetic material, adenine, guanine, cytosine, uracil will be present? That's nothing but the RNA, isn't it? RNA. And how many strands will be present in the RNA? Single strand. Right now, we have discussed this till now. In the first year also we know. Second year in the entire syllabus. If not syllabus, now also I am saying double stranded is the DNA, single stranded is the RNA. So the answer is RNA, comma, single stranded. 2 marks you will get. See how this is very easiest one. This chapter was over. So how many are there? Out 17 questions are there. Out of 17, you will get 2. See how easily we can score the VSQ questions and the marks from them. Okay? Because 9th chapter, 1 question, 1 VSQ. And the next one is the 10th chapter 2. And after that, we'll be having the 7th and the 8th chapter, 1 VSQ. Mostly from the 7th chapter they will give. And 11, 12, we will get 1 VSQ. Very easy. So make sure that you should get good score in the VSQ part, which is easy, beta, easy, very much easy in botany. So the first question in the 11th chapter, that is the biotechnology principles and process. This is not that much important. Define biotechnology, they have said. So what is meant by biotechnology? Biotechnology is the scientific manipulation of biological organisms, especially at the molecular level, to produce new products for the use of for the benefits of the human beings are called as the biotechnology. Okay, the scientific manipulation. The next one is the molecular. What are molecular scissors? Why are they obtained? Where are they obtained from? So the molecular scissors means the restriction endonucleases. The restriction endonucleases which cut the DNA at the sticky ends. Which cut the DNA at the sticky ends, they are called as the molecular scissors. Okay. The molecular scissors are the restriction enzymes, restriction endonucleases, you can say. Restriction enzymes are restriction endonucleases. Restriction endonucleases which will cut the DNA at the specific locations are called as the molecular scissors. Okay. And these are obtained from which? These are obtained from the bacteria. This can be obtained from the bacterial organisms. Those are the molecular scissors. And the next one is name any 2 artificially restructured plasmids. Name any 2 artificially restructured plasmids. So in this, 3 are there. That is the PBr 322, PBr 322, PBr 322, PUC 19, and PUC 101. These are the 3 artificially restructured plasmids what we are having. So we have to write these 3, PBr 322, PUC 19, PUC 101. Okay, done. Next one is what is EcoR1? How does it functions? EcoR1. EcoR1 is the restriction enzyme. EcoR1 is a restriction enzyme. EcoR1 is a restriction enzyme which is obtained from the bacteria Escherichia coli. Escherichia coli. Okay, so this is the EcoR1 is obtained from the bacteria Escherichia coli. So what it will do? It recognizes the palindrome. It recognizes the palindrome having 5' to 3'. That is the GAA-TTC. That is the palindrome which is running from 5' to 3' direction. Okay, GAA-TTC. It recognizes this palindrome and cuts the DNA between G and A. Okay, that is making the sticky ends for the rDNA technology. What is its function means this one. Now comes what are the cloning vectors? Cloning vectors not that much important. We'll go with what is recombinant DNA? Recombinant DNA means the hybrid DNA that is formed by the fusion of hybrid DNA that is formed by the fusion of DNA with the desired gene with the vector carried the desired gene to the host genome. Okay, when a hybrid DNA is formed by joining the desired gene to the vector's DNA. Hybrid DNA formed by the joining desired DNA to the host or vectors DNA. Vectors DNA. Okay, so that is called as the recombinant DNA. So this joining was done by the enzyme ligase. Joining was done by the enzyme ligase. Okay. And this type of rDNA is also called as the recombinant DNA or chimeric DNA. Recombinant DNA or chimeric DNA, rDNA or chimeric DNA. Okay. Now comes the next question. What is palindromic sequence? What is palindromic sequence? Palindromic sequence means the DNA sequence, the nucleotides when read from right to left and left to right in the same manner are called as the complementary nucleotides. Otherwise, the palindromic sequence, not complementary, sorry, palindromic sequence. Okay. The two strands of the DNA which are having the nucleotides the same from the right to left and the left to right are called as the palindromic sequence. And this palindromic sequence is going to be recognized by the specific restriction enzymes. Okay, and they are having the specific restriction sites on them for the specific restriction enzymes. Example, just now we have given GIA-TTC, which will make the cut between G and A by the echo R1. Same thing we can say the example over here. Okay. And what is the full form of PCR? How is it useful in biotechnology? Full form of PCR, polymerase chain reaction is the PCR, polymerase chain reaction. Okay, and how is it useful in biotechnology? To making the multiple copies in a less time period. That is useful. The PCR technology is used for making the multiple copies in a less time period of the desired gene. That is called as the PCR technology. Okay, this is useful in the DNA process, otherwise in the biotechnology also you can write. And what is downstreaming process? Downstreaming process means separation and purification of products, separation and purification of products after the completion of the biosynthetic stage in the rDNA. After the rDNA formation, the separation and purification of the products can be done by the downstream processing, so that it will be easy for doing the release the products in the market. That's the one. And how does one can visualize DNA on an agarose gel? So this one, the DNA fragments gets the darkly stained by this agarose, by the ethidium bromide. Ethidium bromide. Okay, DNA molecules the DNA fragments were darkly stained with the ethidium bromide in the gel electrophoresis machine. With this stain gives the, otherwise imparts, you can write anything, gives the DNA fragments as the orange color, and under the UV light we can see them as the darkly stained, otherwise the lightly stained orange colored strand. That is more in number and less in number respectively. So these are all things can be done on the agarose gel, that is present as the base platform in the gel electrophoresis machine. Okay, that is the one. Separation of the DNA fragments can be visualized only after staining the DNA with ethidium bromide followed by UV radiation. We can see the bright orange colored bands on DNA. That is the agarose gel. Okay. How can you differentiate between exonucleases and endonucleases? Exonucleases means these are the enzymes actually. Okay. What they will do? They will cut the strand at the end. That is the exonucleases. Exonucleases are the enzymes which cut the DNA strand at its end to make the blunt end. And endonucleases. Endonucleases are the ones which will cut the DNA strand at two different positions. Okay, so that it will give the sticky ends. Will give the sticky ends. That's it. So like this, we have to finish it off the VSQs from this chapter. And we will see in the next chapter. I will go fast so that I can wind up this chapter also. Okay, so tomorrow, 10 minutes. 10 minutes are there means I can wind up. So in this, the very first VSAQ. Give the different types of crygenes which are controlled by the proteins encoded by, okay, by these genes. So in this, crygenes are, the Bt toxin is encoded with a gene called as the crygene. The crygenes are of three types: Cry1AC, 2AB, and 1AB. Okay, so Cry1AC controls the Cry1AC and 2AB. You can write like this: Cry1AC and 2AB controls cotton bollworm infections. And Cry1AB controls corn borer infection. That's it. This is the first question. Now comes the second one. Second question. So, can a disease be detected before its symptoms appear? Explain the principle involved. Yes, without the disease, the symptoms can appear first of all. Okay, so that is the, yes, very low concentration of virus and the bacteria can be detected with the amplification of their nucleic acid through the PCR technology. We can easily confirm the infection when that is not a disease also. Okay, what is GEAC and what are its objectives? GEAC means the Genetic Engineering Approval Committee. Genetic Engineering Approval Committee is the GEAC. And this is an organization that was undergoing with the Indian government. Okay. And what is the main objective of this? It makes the decisions regarding the validity of the genetically modified research organisms and the safety of these organisms for the public people, for the public. Okay, so that is the GEAC objectives. And in the next one, name the nematode that infects the roots of tobacco plants. Name the strategy adopted to prevent this. The nematode which infects the roots of tobacco plant, that is the Melodigine incognita. Melodigine incognita. This is the nematode that infects the tobacco roots. Okay, so what is the strategy adopted to prevent this? So what we will do, we will control this infestation with the RNA interface. This infection can be controlled with the RNAi gene, which makes all the eukaryotic organisms cell defense mechanism will be done with the RNAi gene. What it will make? It will silences the mRNA. It silences the mRNA. Silences the mRNA for the translation process, so that for the complementary RNA molecule. So the translation process is going to be stopped. No further amino acids or enzymes are released. So the infection cannot spread. That's the one what we have to say here. Okay. So that is the one. And the next one, which type of variety of Indian rice has a patent filed by USA company? Which company, which USA company filed its patent on which type of crop of India? There is the basmati variety. Everyone knows this. We'll eat biryani, basmati rice. Okay. And the next question, What is green revolution? Who regarded as a father of green revolution? Green revolution means a drastic increase in the food production of the due to the plant breeding is called as the green revolution. Okay, and this was the first father of green revolution is Norman Borlaug. Norman Borlaug. B-O-R-L-O-U-G. Borlaug. So, like this, if you write, green revolution means drastic increase of the food production by the plant breeding process. It's called as the green revolution. Norman Borlaug considered as the father of green revolution. That's it. With this, we have finished the VSQs part from the 9, 10, 11, 12 chapters, and the 7 and 8 were also over. Now we have to go only with the few chapters VSQs, I think so. Okay, so we will see tomorrow with the more number of VSQs, and stay tuned to the same channel with the in the same time to go with the successfully completing the second year botany in this supplementary exams. No worry for you to go for the think that whether I will pass or fail. You have to pass, no chance, no option left out. Okay, right? Prepare well, whatever I have discussed now, the VSQs and the elective and the SQs till now. So, okay, right, students, will stop the class for today's here.